The overall goal of the following experiment is to analyze the conformational flexibility in proteins and changes thereof in response to environment, ligand binding, or protein-protein interactions. This is achieved by pre-incubating the protein of interest under different conditions which are suspected to influence the conformation of the protein, such as ligand binding. The reaction mixture is then diluted into deuterium oxide, incubated for different time intervals, and subsequently analyzed by liquid chromatography mass spectrometry to determine the degree of deuteron incorporation into the peptide backbone amide groups. Because protected areas have decreased incorporation of deuterons, comparing the peptide spectra of the experiment in the absence and presence of a binding partner reveals a shift of the centroid of the spectra due to the weight difference between proton and deuteron. Regions that exhibit prominent protection after addition of a binding partner are potential binding sites. Depending on the sequence coverage and the availability of overlapping peptides, binding sites can be narrowed down to a few amino acids.
This method can help to answer key questions in the protein folding and the protein dynamics field, such as how proteins fold, how they unfold, how they react to physical and chemical impacts like temperature increase or heavy metals, how ligand binding and protein-protein interactions can influence the conformation of a protein. It is also very important for the development of protein drugs, for verification of proper folding, and for general quality control.
Generally, individuals new to this method may encounter several difficulties. If the protein, for example, is not well digested,